that examines the role of symbionts in mitigating thermal stress of large benthic foraminifera. And let's start. So, next slide, please. Can you pass to the next slide?
Yeah, sure. Actually, voice is breaking. Anyway, I am on the next.
We didn't understand nothing for your previous explanation. Could you please start from the beginning? The voice was not clear. Thank you.
Yeah, no?
Okay.
My voice. Oh yeah, okay. So all right. I've just said that today I will talk. Said basically I will just talk about how the role of symbiont in mitigating thermal stress of foraminifera, and then I've asked to move the slide. So. Hello.
Yeah, I moved it.
I can see it. Let me just connect from my phone. Maybe it's the easiest thing. Sorry. Can you hear me? Or I just don't know if I. Yeah. Okay. So let's try. So just signal to me if you can't hear me well. But I'll just try to start. So my research in general deals with the effect of future warming on calcifying organisms. Better?
Better.
Yeah.
Better.
All right, great. So the theme that keeps coming up, whether I work on foraminifera or coccolithophores or algae, is the relationship between photosynthesis and calcification and the role that it has in mitigating environmental stress. And this is, of course, important because marine calcifiers are ecosystem engineers that so while larger organisms like algae and corals build habitats to support high biodiversity, foraminifera are really important because they contribute to
Danna, I'm sorry, Danna, but we can barely. Dan, I'm sorry, but we really do not understand any word.
Danna? Hello, Danna?
I still see the first.
Yeah, Danna, we can't see, we can't hear you actually. So I can move the slide, but the thing is that we can't hear you, Danna. So
yeah, no, I don't think this will work. Do you want me to try to reconnect?
yeah, maybe you can try.
can't hear me, yeah, if you can't hear me, then the issue. I want to try and reconnect if that's okay. I'm just gonna hop in and out and see if it gets better. Can you hear me now?
Yeah, so maybe with the permission of
Can you hear me now?
yeah, I can hear you. Yes, I can hear you.
Okay, I've logged in through my phone, so I'll just try to do it this way if you can hear me properly. Can you hear me?
Yes.
Guys, let me just put some. Okay, let's try it this way. Hopefully it would work. So could you hear what I've said before or not really?
No, not really.
All right, let's try it again one more time. Again, I apologize for the poor connection. Hopefully this will sort it out. Yeah, so let's just start from the first slide of the presentation, because I guess we're running out of time, so I'll skip the intro. Future warming and calcifying organisms. And the theme that keeps coming up, whether I work in coccolithophores or foraminifera or algae, is the relationship between photosynthesis and calcification, and the role that it has in mitigating environmental change or environmental stress. And this is, of course, important because marine calcifiers are ecosystem engineers that physically change and build the environment that they are in. So while large organisms like algae and corals build habitats that support high biodiversity, foraminifera are really important because they contribute to the cementation of reef structures, and they produce sand that maintain shoreline as sea level rises. Okay, next slide, please. All right, so foraminifera, they're not only important as carbonate and sand producer, they are also really interesting in the context of their very high resilience to heat stress. Now, some modern species have been shown to calcify up to even 40 degrees, and during past events of extreme climate, the fossil record demonstrated their exceptionally high resilience compared to other groups of organisms, and that indicates their potential role in a warmer future world. Next slide. Okay, so it was suggested that the high thermal tolerance of large benthic foraminifera is related to the high flexibility of algal symbiosis that includes, for example, diatom, dinoflagellate, and rhodophytes. And just before I continue, I would just like to explain or mention that to avoid from saying symbiont-bearing large benthic foraminifera many times during the talk, I'm just going to call them LBFs. Anyway, the algae symbiont photosynthesizes and provides sugars and other organic compounds to the host while receiving UV protection and metabolites from the host itself. And the less the host is dependent on the algae for acquiring energy, the more it needs to rely on the bacterial community as a food source. So this extra energy that they can get from the algal symbiosis allows them to deal with environmental stress and continue to calcify. Right, next slide. So here you see an example of why we think the algal symbiont supports the host. In this study, we quantified the change in calcification and in photosynthesis in omphisogynal lobifera under different temperatures. And what we saw is that calcification is inhibited at 15 and 35 degrees, so basically under cold stress and also under warm stress, with optimum calcification rate at 25 degrees, while photosynthesis remained constant between 15 and 32 degrees, and only decreased to 35. So basically, and you can see this in this schematic sketch that I've made here, basically the optimal temperature range for photosynthesis is much wider than that of calcification. So theoretically, the symbiont can support the host outside of its optimal range by providing the product of photosynthesis and providing energy. All right, next slide. So to try to understand the mechanism behind it and to see if this is really the case, that the symbiont supports the host because it has a wider temperature range, we conducted transcriptome analysis on samples from the 15 degrees, the 25 degrees, and the 32 degrees treatment. And what we saw is that cold and warm stress have completely different mechanism of handling, or like the algae and the foram have very different mechanism of handling cold and warm stress. And basically what you see here is that a lot more differential regulation is involved in dealing with cold stress compared to warm stress. But let's move to the next slide. So to remain on the focus of the role of algae and photosynthesis and how it supports calcification, I now focus on genes that are related to photosynthetic activity. And briefly, what you can see here is that both in warm and in cold stress, genes are differentially regulated, despite the fact that the performance in terms of oxygen production is the same. And basically what this means is that there is a relocation of cellular resources by the algae to photosynthesis at the expense of other processes. All right, let's move to the next slide.
बाद में कंफर्म कर लेंगे। Anyway, तुमने बस सर को आइडिया लग गया होगा, नोट कर लिया।
So when we
टेंशन लेने की जरूरत नहीं है। ये गलती को ऑलरेडी
Sorry, you've muted me. Can you hear me? Yeah. All right. So when I can hear someone talking. Could you mute? Hello? Yeah, should I keep going?
Yes, go ahead.
All right. So when we look at other LBF species, we see that they show different trends. So in Amphitragina, as I've shown before, you can see here on the graphs on the left, it's a different experiment, but we see a similar thing, wider temperature range for photosynthesis compared to calcification. And next to that, you can see the species Soraitus orbiculus. You can see the trend here is exactly the opposite. So the calcification remains high throughout the range of 25 to 35 degrees, but the photosynthesis actually decreases to negative values at 35 degrees. So in this case, actually the host is more resilient than the symbiont. And here it's not as clear, or it's actually clear that the symbiont could not support the host, unlike in the other species. And the two panels on the right, I just brought it to show that it doesn't have to be not synchronized between the symbiont and the host. You can see that both calcification in the top graphs and photosynthesis in the lower graphs are actually the same for the species, but one for operculine. So I think the problem is that every time you mute everyone, you mute me as well. Hello?
Yes, Danna, please.
Yeah, so every time you mute everyone, you mute me. And I think, I'm not sure how much you hear.
No. We are hearing you. Please, please.
Okay. Okay, I'm not sure how much you heard. So let's move to the next slide, and I'll just try to recap from here. Okay, so basically, because different species do different things in terms of host and symbiont, I've decided to look into a very resilient foraminifera species, and that species is called Pararotalia calcariformata. And how do I know that this species is so resilient? Because we find it in a thermally disturbed area in the northern coast of Israel. Now, this thermal disturbance is created by an electrical power plant that pumps water to cool the turbines and then discharges it back into the sea, creating a thermal gradient of about 1.5 kilometers. And what we did is that we've monitored the environmental parameters and the foraminifera populations along this thermal gradient for about 15 months. In the graph here, you can see the temperature that we've recorded throughout the study. Basically, you can see in red, the warm part, the warm station is consistently much warmer than a control station marked in blue. And we're talking about an average of about 5 degrees, but a maximum of 10 degrees. So it is much warmer. And all the other environmental parameters that we have monitored were similar. So we know that any change in foraminifera population would be the result of, or probably the result of that change in temperature. And I'm not going to go into all of the detail of the study, but basically the important thing is that in the warmest part of this heat disturbance, there were only 4 species that were able to live there, and only one of them had symbiont bearing, and that is the Palaeogene that I'm talking about. Let's move to the next slide. So some more evidence to the fact that the species is really resilient. So here you can see the numerical abundances of the 4 species that live in the thermal pollution. These are the red bars, and the black diamonds are the monthly average temperature that they were exposed to. And basically what you see is that all of the species, except for Palaeogene, which is the bottom right, they abundances between April and May when temperature is about 30 degrees. And only Palaeogene continues to show high abundances all the way through June. So this was the first evidence that indeed this is a very resilient species, and definitely more resilient than the other species who also occupy the plume. So let's move to the next slide. All right, so here what I want to show is basically that, in fact, it's not just resilient enough to survive there, it's actually resilient enough to calcify under the very high temperatures. And what you see here in these graphs, so basically what we did is that I took the samples, the shells that I've collected over a year, and I've analyzed the magnesium calcium in the last chambers. And then using a calibration that I did with a laboratory experiment, I've calibrated the magnesium calcium value to the actual temperature of calcification. And I won't go into all the details because I think we've lost a lot of time here. But basically what you can see is that the gray part in the graph is the actual temperature that the foraminifera was exposed to. And you can see that the black diamonds that are representing the calcification temperature, they're not lower than that gray, the gray area that represents the actual temperature, meaning, yes, this species can actually calcify even at 36 degrees, which is the highest that it was exposed to in the time. All right, let's move to the next slide. Okay, so now we know that the species is extremely resilient, and this is why it's the target species for the studies that I'm going to show, but also for other studies along the way. And this is why in 2018, Christiana Schmidt et al. Have published a study characterizing the symbiont community of these species in different locations around this thermal disturbance. And they've used 4 locations. The upper bar that you see here is actually a control station, and then the one beneath it is a station along the thermal gradient, but not quite at the core where it's the warmest, but more to the edge of the heat plume, where temperature are about 2 degrees higher than normal conditions. And the 2 stations that they've examined that you see here on the bottom are actually a little deeper at about 5 meter water depth, but they are also affected by the warm water disturbance. And what Schmidt has shown is that basically, if you look at all the locations that she's examined, she found about 17 species of diatom symbionts. But from all of the locations, the same species was actually dominated in all of the specimens. And what they have concluded is that the diverse internal symbiont community of Palaeogene provides potential for it to shuffle the community of symbionts, and therefore use the more adapted or the more appropriate symbionts to create the high resilience. And that was really interesting, but I think what was still lacking at this point was how exactly do they do it? How do they get these more resilient symbionts to be more dominant? So, okay, next slide, please. Yeah, so basically there are 2 kinds of mechanisms in which this could happen. And this is something that we know from coral literature. basically there are two options, symbiont shuffling and symbiont switching. When we talk about shuffling, we basically talk about strains of symbionts within the internal community of the host that, according to environmental conditions, can reproduce faster and become more dominant in the internal population. The other option is switching. In switching, it's not about the internal population, it's about what's available in the surrounding water. So basically, the host can expel the symbionts that it has and pick up a more appropriate or a more adapted species from the water, and then basically culture it into being dominant and using the products of photosynthesis to support the process of calcification. All right, next slide. All right, so what do foraminifera do? Is it symbiont shuffling or symbiont switching? This was something that I was really interested in. And so when I got to the group where I work today in University of Oxford, and I've started dealing with isolated or cultures of just algae, not of foraminifera, I thought it was a really good opportunity to try and look into this question of how do algae, symbiont algae supports the host. And to do that, I've focused on two species that I've already mentioned before. And this is a Palaeogene that you see here on the right. It's a very resilient species that can calcify up to 36 degrees. And then on the left is the second species of holobiont. And this is a very, very common species with abundances in many, many different places across the globe. But as you can see here from another study by Schmidt, you can see that the symbionts as a community are much less resilient for Amphistigena than for Palaeogene. You basically see that in the summer populations, there is no change in symbiont performance for Palaeogene, and in the winter, there is a small decrease at 36 degrees, while in Amphistigena, there is a huge decrease at 36 degrees, and even before so. So this is why I've chosen two species. One is more resilient than the other, and we suspect that this is related to their symbionts because we see, according to these graphs that are on the screen now, that they perform much better under high temperatures. All right, next slide, please. Okay, so basically what I did just to start off is that I took these two species and I did a very simple temperature experiment. I've exposed them to 25 degrees, 32 and 35 degrees, and I've exposed them for 30 days, just replacing the water so they have enough nutrients and everything that they need. And then after 30 days, I've measured the oxygen that they produce, so basically their photosynthetic rate. And also I've analyzed the community structure of their symbionts after 30 days exposed to these temperatures. So let's start just by looking at the oxygen production. Basically, these are the graphs at the left. And basically what you can see is that, yeah, both species have higher rates or they produce more oxygen at 25 degrees compared to 32 and 35. But you can see that for Amphistigena, the reduction is so high, the reduction is so massive that they are actually, the net photosynthesis is zero. So there's no oxygen produced and there's no products of photosynthesis that can be 2 degrees. It's not as massive. There's still positive production of oxygen. So it could still, if it has a role in providing energy to the host, it could still do that. Okay, so that was quite interesting and still supporting our argument that thermal tolerance might come from the symbionts. And in this case, we're only talking about diatoms. These are two species that has diatom symbionts. So now looking at the symbiont assemblages after 30 days of exposure to different temperature. And also something that I haven't mentioned that I just want everyone to know. So 25 is the optimal temperature for both of these species. 32 degrees imposes some stress on Amphistigena, but not on Palaeogene. And at 35 degrees, Amphistigena is heavily stressed in a way that it doesn't calcify anymore. And Palaeogene seems to be absolutely fine in this temperature and continue to calcify. All right, so how does this graph work? There are three circles. The inner circle is the family level, the middle circle is the genus level, and the outer circle is the species level. The color on the graphs represent the family level of the symbionts, of the diatoms, and the number that I've written on the outer circle is the percentage of a specific species. So this is to give you an indication of the diversity, of changes in the diversity. All right, so what do we see here? We see that starting with Amphistygina, we see there is a major change in symbionts. So basically, and this has to be shuffling because these are synthetic water that I've put in the experiment. There are no other algae for them to pick up. Whatever change that they make, they have to do with their own symbionts. And what we see is that indeed there is a large change in the level of family. So you can see in 25 degrees, it is dominated by Fragilari. So this is the family marked in green. And pretty gradually, when it goes to 32 and 35 degrees, so as we elevate thermal stress, the dominant family is changing to the family marked in red. Okay, when we look at the more resilient species, the one at the bottom, these are the three circles that are basically marked in purple. And because they're marked in purple, you can tell that they are all dominated by one family, no matter what temperature they're exposed to. And yeah, in 25 degrees, there was some presence of the family marked in red, but you can see very clearly from the beginning, even at 25 degrees, it is dominated by a specific family of symbionts that is only present in small amounts in all treatments of Amphistygina. And I think this is the really interesting thing. So Amphistygina does have not only this family, but the specific species that is dominated in Palaeogene, but for some reason, it did not shuffle it to be more dominant in the thermal stress treatments. So we can't say from this why. This could be either related to their flexibility, their ability to use whatever symbiont that they have, or that it could be that the symbiont isn't actually producing the thermal tolerance. Okay, so to further look into that, I've decided to do another experiment. Can we move to the next slide? Thank you. Yeah, and what I wanted to do is to look at each symbiont separately, because up until now, we only look at the symbiont as a community. So all the species that are in the host, what do they do together? And I really want to see, okay, are there key species that are more resilient than other? And could they possibly be what supports? And is that one species that is dominant in Palaeogene, but quite rare at Amphistygina, could that be the species that provides thermal tolerance? So what I did is that I've isolated the symbionts from Amphistygina and from Palaeogene, and here you can see the strains that I've worked on. And this is not the full diversity of symbionts within these species, but these are the symbionts that I have worked on. And the reason that I'm showing this is that I want you to see. So the black lines show which symbionts came from which foraminifera, and you can see that there is an overlap with one strain between Amphistygina and Palaeogene. Included the black line for perculanic. So this is just to show you that there are symbionts that are also in common with Operculana, and the reason that I show this is because Operculana is also a very resilient foraminifera, resilient to temperature, of course. So this would indicate to me that these strains might be highly resilient. So after isolating them and creating monocultures with just one strain of symbionts in each culture, I wanted to do a temperature experiment to examine their thermal tolerance. And because I suppose most of us are foraminifera experts and not algae experts, I'll just briefly explain how this experiment works. Basically, we inoculate from the culture to a bottle with seawater, and we start monitoring the amount of cells. We count the cells in the bottle in each day. And this is how we track the growth of the culture. We track the growth of, or the reproduction of the algae and how they multiply, how fast they multiply. At first, in the first days, nothing happens. They stay similar. And the length of this phase, which is called the lag phase, is actually indicative of how much stress the culture is under. So if there's no stress, they will immediately start growing. If there's stress, they will need time to adjust and basically acclimatize themselves to the new condition, and then they will start growing. And this is when they will go into the second phase, is that you see in the sketch in the bottom part of the slide. When they start growing, they grow in exponential phase, which thanks to COVID, we all know what that means, so I don't need to explain it. Basically, they start growing very fast until they deplete the nutrients in the water in the bottle. At this point, their growth will stop and then crash. So this is what happens during the experiment. So to maintain the experiment through more than these few days that this takes, during exponential change, we actually take a little bit of culture and move it to new water with nutrients. So this is how the culture can keep going. And the two parameters that I've examined is, so it's the length of the lag phase that indicates stress immediately as the culture starts, and also the growth rate. So this is something that we measure during the exponential phase of growth. All right, so let's move to the next slide and see the results of this experiment. Yeah, okay, so here we start with the lag phase, and we can see that for most of the diatom strains that I've examined, we have a similar length that is lower than 5 days, and there are a few cases in which the lag phase were longer, which means it was harder for the culture to continue its normal growth under these conditions. And the color on the graph shows the amount of time that the experiment was going on. So if it's pink colors, it's just the beginning of the experiment, and if it's green color, it's further down the line. And the experiment lasted 40 days. So the dark green is about 40 days. And what you can see here is that for the two pink dots that are higher, that have higher leg lengths, this only happened in the pink. So after a few transfers of the culture, it could actually acclimate to the temperature, and then it grew fine. And then you can see this very high green points of the last Amphistogina diatom strain, and you can see very long, very long leg phases that indicates these species might have really had to struggle to grow at 32 at 35 degrees. So they did eventually start growing, but it took some time. Now let's move to the next slide and look at the growth rates. So here you can see the growth rate. So growth rate would change according to the stress level of the culture. Basically, if there is stress in one of the treatments, then we expect that the growth rate will be much lower. And what we see here is that all of the Amphistogina have similar growth rate under the temperature that they grow in, but they either grow at 25 or at 25 and 32. They don't really grow at 35, other than this one case where it actually took almost 40 days for it to just start growing. So it does grow at 35 degrees, but it obviously experiences a lot of stress. The optimistic thing is that it does show some potential to adaptation, but that's for a different talk. And when we look at the symbionts isolated from the Palaeogene, we see that two of them only grow at 25 degrees, but one of them actually grows at 25, at 32, and also at 35 degrees, and at similar growth rate, which means it is indeed more tolerant to high temperature compared to the other symbionts from both species. So my guess would be that if there is a strain of diatom algae that provides the foraminifera host, Palaeogene, with thermal tolerance, then it would definitely have to be this species. So to look into that, can we move to the next slide, please? Thank you. I've decided to do another experiment. This is a switching experiment. So basically, I took Amphistogina samples that were exposed to 32 degrees for about 2 months. So they were partially bleached, but they were definitely still alive. This is what their symbiont community looks like, and this is exactly what we saw before. So it's dominated by a genus called Nichia, but it has also symbionts from Fragillaris family, which was the dominant family at 25 degrees, and also a little bit of symbionts that are dominant in Palaeogene, the very tolerant foraminifera species. And what they did is that I took them when they're partially bleached from 32 degrees, and I've divided them into 4 groups. And can we move to the next slide? Thank you. Okay, so I've divided them into 4 groups. 2 of the groups I've put in 25 degrees, and 2 of the groups I've left at 32 degrees. And every few days, I've changed the water, the seawater that they were in, and I've also introduced the symbionts of Palaeogene. So the 3 symbionts that we now know, 2 of them are not so resilient to temperature. They only grow, the culture only grows at 25, and 1 of them also grow at 32 and 35 degrees. But I've continued to introduce living diatoms, so it's not like they died off during the experiment. They were always present there. And it went on for another 2 months or so. And after 2 months, I've stopped the experiment. I've taken the samples out, and this is to demonstrate what I've seen. So basically, I saw that at 25 degrees, both with the introduction of symbionts and without the introduction of symbionts, there was a recovery because some specimens, as you can see, were no longer bleached. Now, under 32 degrees, in the control where no symbionts were introduced, you can see there's quite heavy bleaching, Now, the specimens, most of them were still alive. They were still pseudopodal activity, so I know they were alive, but they were definitely bleached to the point that they were white. We could not see any symbionts in them. However, in the 32 degrees, when I did introduce the symbionts, you can see the strong color. So it's very obvious that they have picked up or they were able to recover and maintain their symbiont population. And so to me, even without going further, this was an indication that yes, there is something happening when you introduce the tolerance symbionts from Palaeogene. When you introduce it to thermally stressed Amphistigena, it is definitely able to recover thanks to that. And this is the difference between the control 32 treatment and the symbiont introduced 32 treatment. Right, let's move to the next slide. So here we see the symbiont community in each of these treatments after the introduction of symbionts and in the control. And we saw something that I found very interesting. We saw that under 25 degrees, both with and without the symbionts, the community of the symbionts looked exactly the same. And it looks the same as in the original experiment when I've exposed Amphistigena to 25 degrees. It's heavily dominated by the family Fragillaris with some presence of another family, but it doesn't seem to be related to the introduction of symbionts because it's similar in the control and in the actual treatment. Now, looking at 32 degrees, we can see that the bleached specimens are dominated by diatoms from the genus Nichia, which is completely different from the diatoms that are dominant in the treatment of 32 degrees with the introduction of symbionts. And this difference means that, yeah, it's not the same process. Something has happened there. They have either shuffled or switched to a more tolerant symbiont, which was then able to reproduce and provide this beautiful color of the Amphistigena. All right, let's move to the next slide. And I'll just try to summarize because I realize because of all the delays in my connection, we're running out of time. So this is all, as you can see, quite preliminary, but I think it's quite obvious that we can say at this point that while shuffling doesn't necessarily provide thermal tolerance to the host, the potential of switching, and it doesn't have to be switching to the actual algae, it might be something that comes along, like, for example, bacterial symbiosis, but something is definitely going on that the foraminifera is picking up from the water that allows it to enhance the thermal tolerance. And I think that's super optimistic, and it makes me quite optimistic about the future of our warmer upcoming world. And yeah, mostly I want to say I think there's a lot of places to go with this that I intend to go, and mostly I think this is a really interesting approach to explore the symbiosis and how it affects the resilience of the host to different environmental stressors. And also, I think it's a really interesting perspective to see if we can kind of enhance the thermal resilience of foraminifera. All right, and I think I'll end with that. So thank you. And you can move to the next slide. And I just want to say, if anyone is working on symbiont-bearing calcifiers or want to take part in this, and you would like to send me your foraminifera for me to isolate the symbionts, then I think that would be a great effort, and it would allow us to continue and explore this interesting path of creating more tolerant foraminifera. Right, thank you.
Thank you so much for Danna for this nice presentation, though we had a very, the starting was a little bit problematic, but yeah, it's really nice talk. So since we are already out of time, we may actually have 1 or 2 questions. So anyone, there is any question? Otherwise, obviously, you can talk to Danna by through this email. So any question? So I think people are thinking about this running out of time thing. So
yeah, sorry about that.
No, it's okay. So yeah, so I think I can request all the participants, since we are running out of time, that I'm very sure that there are lots of questions, because I'm also having few questions, but only because of time, I am very sure that I'm going to ask Danna these questions through email. So thank you, Danna. Thank you for your this wonderful talk. And I'm just stop sharing. Again, anyone want to ask one or two question here? We can accommodate to one or two question.
You can see the chat box, Urvashi.
Chat box, I am. Yeah, there is one question. I am interested in the method of isolating algae, especially when you said few slides ago. How did you monitor cell numbers? Yeah, so the question is, how did you monitor cell numbers? Danna,
right, so yes. So to monitor cell numbers, we use a flow cytometer. So you basically put the culture in the flow cytometer, and it gives you a count of how many particles are in there, and it gives you a read of the fluorescence, so you know that you only read live cells, and you just do it every day, so you know how the culture is growing. Does that answer the question?
Yeah, okay, so she thanked you. So I'm just stopping sharing your PPT, Danna. It's all right, because I need to share another one. Yeah,
yes,
okay, okay. So, yeah, thank you, everyone. Thank you for participating in today's seminar. So we are having our next seminar on 20th February at 14 CET. And it is going to be, the speaker is Helen K. Coxell from Stockholm University, and she is going to talk on planktic foraminifera in high Arctic past and the present. So we will be looking forward to all of you to participate in our seminar. Also spread the word that the next speaker is Helen Coxell, and she is going to talk on planktic foraminifera. So thank you to all of you for participating in our this seminar. Thank you.
Thank you very much. Thank you very much to everybody. Thank you.